Okay, excited to get the flash talk started. So my name is Tavor Baharav. I'm a second-year postdoc at the Eric and Wendy Schmidt Center, and I'm here today to talk about T cells. So through the process of VDJ recombination, T cells recombine their genome in alpha-beta pairings, constituting on the order of like 10 to the 15 different T cell receptors. This massive diversity makes estimating an individual's T cell receptor profile both a difficult and important problem. So traditionally, single cell is used to identify these pairings between alpha and beta chains. However, this is expensive and can't profile too many cells. Bulk, on the other hand, is cheap and can profile many cells, but doesn't give you this pairing information. So together with Jeremy Simon and Rafa Irizarry, I've been working on these kind of combinatorial problems and integrating these two different modalities and beyond just bulk and single cell. And we've developed some new statistical methods, which we're working with Dr. Kathy Wu's lab to validate. So please come by and chat with me at poster 1 if any of this sounds interesting. Thank you.
Hi, I'm Salil Bhate. My work is motivated by the question of how does the genome sequence encode phenotypic structure across levels of complexity. So we have experiments now that measure genotype-phenotype associations from cells to tissues to organisms, and we have really good algorithms that predict these associations and learning from functional genomics and other kinds of data. So are there any universal principles that let us derive measured genotype-phenotype associations akin to natural laws in physics? So the computational task is to recover measured genotype-phenotype associations from just one phenotypic measurement, for example, one tissue or protein sequence, and the reference genome. And that's without annotations, genotype-phenotype covariation, or mechanistic information. And what's surprising is by searching for structure-preserving codes, that is, codes that assign genomic loci to phenotypic units preserving similarity, we can actually solve this problem and recover measured associations across scales. So yeah, come check out my poster number 2.
All right, thank you, Salil. Can I have Xingjian up next?
Hello everyone, my name is Xingjian Chen and I'm a postdoc in Jiangshu lab at MGH. Thank you for the great opportunity to share our work today. So we are building a DALI for biology, basically a gene-to-image foundation model called GenVinci. So this work is supervised by Professors Colin Strauss, Aviv Regev, and Jian Shu. So GenVinci can turn single-cell gene expression into different kinds of image modalities, including H&E images, DEPI, immunofluorescence, neuromorphology, and electron microscopy. And just like DALI, that can generate images from text. So GenVinci aims to be a multimodal view of cell states. If you are interested, please come to our poster number 5. Thank you.
Thank you. Can I have Gillian up next?
Hi, I'm Gillian. I'm a PhD candidate with Ben Raphael in the Department of Computer Science at Princeton. And I want to talk to you about LeNail. So one recent approach to studying cell development leverages dynamic lineage tracing technologies, which involves incorporating a lineage tracing system, which is a genome editor that's going to accumulate edits at specified target sites in a cell. And then that cell divides, and you can use that to reconstruct the cell division history. So the computational problem there is taking those edits summarized in a character matrix, which I have on the left in the inputs. Summarized in a character matrix, the computational problem is to infer the cell lineage tree. So LaML, or lineage analysis via maximum likelihood, infers the maximum likelihood lineage tree and also parameters like time-scaled branch lengths and missing data rates. And LaML infers, this is on the right-hand side now, LaML infers more accurate tree topologies on simulated data, but we also see evidence of this in real biological data. And more importantly, more recently, as of the last 2 months, we've re-implemented some key inference steps, and now we have the most scalable method compared to methods of comparable accuracy. So this is very exciting. So if you work on any lineage tracing or are just interested in cell development, please come talk to me. I'm at poster number 6. We also have a new method that's coming out soon. This is LaML Pro, which adds modeling of observation error. So thank you so much.
Thank you, Gillian. We have Yasha up next.
Hi everyone, my name is Yasha Ektefaie. I'm a PhD student at Harvard Medical School, co-advised by Marinka Zitnik and Maha Farhad, and I'll be talking about my work in Fleming. Tuberculosis is one of the leading causes of death worldwide by infectious disease. And although we have antibiotics to treat tuberculosis, every year those antibiotics become less effective due to drug resistance. We need new antibiotics now. However, developing these drugs is timely and very and costs a lot of money. And so to address this, we developed Fleming, an AI agent for developing antibiotics for tuberculosis. Fleming is basically a large language model that we've given a bunch of tools that will help it to design a drug. For example, a generative model to generate an antibiotic and a discriminative model to predict, given a small molecule, whether it inhibit the growth of TB or not. And we show in our work that Fleming is able to mimic the reasoning of medicinal chemists and potentially design new antibiotics to treat TB. If you're interested in hearing more, please come by my poster 8. Thank you.
Up next, we have Wenbin.
Hi, everyone. I'm Wenbin, and I'm a recent graduate from UCLA. My work is about modeling of DNA methylation states. So conventional approach usually cares about the first-order statistics, namely the average methylation level. This will ignore the site dependency of different sites. So here, the information loss is more evident from the toy example where we have the two patterns here, distinct pattern, but they have the same methylation level. So when it comes to generation, the pattern 1 will be more likely if the sites are truly independent. This is not the case in real data, as we observe higher mutual information between adjacent sites than what the independent model predicts. So we propose two approaches to model the site dependency. One is the heterogeneous hit marker model, where the transition probability is modeled as a function of genomic distance. And the second one is to implicitly learn the dependency structure using the bidirectional long short-term memory network. And if you want to learn more about the generative models and also the epigenomics, I would love to talk more at my poster. And here is the QR code if you want to check the code and details. Thank you.
Thank you very much. Next, we have Akhil.
Hi, everyone. So I'm Akhil coming to you from UT Austin, and I'll present some work on matrix completion for biomedically inspired missingness patterns. Here on the screen, you see certain kinds of matrix structures which arise in settings that people in this room care about, such as serum-virus interactions, metabolic networks, and gene expression. In one sentence, what we do is combine transfer and active learning in a statistically optimal way for these kinds of problems. More precisely, we have a scalable and robust algorithm which both tells you what matrix entries to select, so this is an experimental design or agentic component, and how to estimate given limited data, which is the transfer learning component. So for bioinformaticians, the takeaway is that you should in fact use and critique these models on your data set. This is very flexible and can be used with several different kinds of side information, which makes it very advantageous for practitioners. And for biostatisticians, what's interesting about this work is that we develop theoretical guarantees and rigorous algorithms, and so we'd love to get your thoughts on those as well. So please come stop by poster 11 and talk with us.
Thank you, Akhil. Next up, we have Khrystofor.
Hello, I'm Chris, a first-year postdoc at the Case Lab abroad. So our work focused on the cultural discovery in a very similar way to what Jean-Philippe discovered earlier today. Thank you. We explored multiple foundational models for molecular representation using the broad prism reporting data as our training data set with the state-of-the-art ChemProp model. Unfortunately, the results were fairly mediocre, but with the graph isomorphism network model, minimal, thankfully, it worked significantly better. So minimal is a graph foundation model pre-trained on millions of molecules. So I think this enables significantly better significantly more meaningful embeddings. So currently, we're trying to arrange the lab validation with a broad center for development of therapeutics. I want to thank our sponsors. This work was funded by the MGH Grants Family Center for Cancer Research and the Human Frontier Science Program. Thank you.
Thank you. Next up, we have Alex.
Hi, my name is Alex. I'm a graduate student for 2 more days in Miriam Hayman's lab. We were interested in characterizing novel transcription factors to rejuvenate old neurons. Inspired by a bunch of transcription factor rejuvenation projects in the field, we wanted to build a perturbation atlas to find potential new transcription factors. So we used a bunch of algorithms to predict transcription factors and the transcriptional effects that they would have. From that, we selected a library of transcription factors that we predicted would be rejuvenative. We did a large in vivo perturb-seq study to validate those. We packaged the ORFs for 156 transcription factors in AAVs. And we generated this large single-cell data set from which we were able to validate the many algorithms that we use to predict the transcriptional effects of those transcription factors. So if you want to hear more about it, come talk to me at poster 14.
Thank you. Up next, we have Prisha.
So T cells are the building blocks of the human immune system, but they're very difficult to predict which antigens they would bind to, which threats they can recognize because of cross-reactivity and data sparsity. So we deal with this by introducing a new related but cheaper task of T cell type prediction. So this multitask approach introduces biological context and allows the model to mirror the way the human immune system actually works and be able to better predict how it would respond. So the model actually uses a shared representation from a TCR representation model, as well as task-specific characteristics from separate classification blocks. So this shared learning really allows for more generalized representations. And when is this shared learning actually useful? If you'd like to discuss this, you can find me at poster 16. I'm Prisha. Thank you very much.
Thank you. Up next, we have Victoria.
Hi, I'm Viktoria Schuster. I'm a postdoc at the Schmidt Center and formerly at the University of Copenhagen. Sometimes we deal with high-dimensional data, but not quite as many samples as we would like. So for this purpose, we've developed a decoder-only generative model that is highly data efficient compared to traditional autoencoder-based models and learns very expressive latent representations. We've applied this to single-cell multiomics data, and because there's no encoder, certain things become quite easy. For example, integrating novel covariates without fine-tuning and in silico perturbations. My latest project has been to improve how we can understand what is learned in latent representations, and for this purpose, we use sparse autoencoders to extract features and automatically annotate them with terms from different knowledge bases. Come see me at poster 17 if you want to understand how this works.
Thank you, Victoria. Up is Jiageng.
Hello, everyone. I'm Jiageng from Brigham and Women's Hospital and Harvard Medical School. Our study mainly focuses on how to leverage large language model to support automatic assessment of cognitive function sector for dementia patients. As we know, each patient has many electronic health records in hospital system. Although these nodes contains rich clinical information, but actually they are also longitudinal, large scale, and clinical domain specific that may be hard to understood by the large language models. So we need to integrate several LM technologies, such as summarizations, such as knowledge enhancement with external source, such as PubMed or Wikipedia. So if you are also interested into improving large language model application in clinical health, feel free to stop by the number 19 to talk more. Thank you.
All right, thank you. Up is Liangying Yin.
Hi everyone, I'm a first-year postdoc fellow at the Eric Edmondson Center. So qualifying and quantify the exact role of individual genes and their direct and total causal effects on complex traits is crucial for a better understanding of the complex biological mechanisms leading to complex traits variations. So we propose a novel Bayes network-based framework that integrates GWAS data and gene expression data from different data sources to construct the gene-gene phenotype causal network. It allows the gene expression data and disease traits to be estimated from different samples, and it exhibits high specificity and precision in identifying causal genes and showed significantly improved causal effects estimation accuracy. And also the identified causal genes in the UK Biobank for the disease traits were significantly enriched for their corresponding targets. So overall, it provides a flexible and a framework for elucidating the genetic basis of complex traits through a system level and causal inference approach. Thank you.
Thank you, thank you. Up next, we have Xinhe.
Thank you. Hi, my name is Xinhe. I'm a graduate student at Harvard University and Broad Institute. Today, it's my pleasure and honor to present to you the AI cyborg system that enables adaptive modulation of organoid maturation. So in this work, we integrate stretchable tissue-like mesh electronics within human iPSC-derived cardiac organoids, allowing for continuous electrical stimulation and recording without disrupting the tissue development. So based on the electrical data, we construct a pseudo-temporal trajectory, a concept adapted from the genomics analytical methods, to represent the organoid maturation functionally. And based on this trajectory, we employ a Bayesian optimization framework with a Gaussian process surrogate model to learn stimulation policies that dynamically adapt to the biological evolving state to achieve optimization with high sample efficiency. This approach embarks a new paradigm for cybernetic control for biological systems that's integrating real-time sensing, learning, and actuation. To learn more about this work, we invite you to visit us at poster number 21, as well as our manuscript on bioRxiv in the QR code. Thank you.
And last up, we have Chase.
Hi everyone, I'm Chase. I'm a first-year postdoc at Broad and Harvard Medical School. And one of the benefits of being last is I see how much time I have left, which is plenty. Everybody did such a great job going, which means I get to take as much time as I want, I think. So what I'm going to reemphasize here is we heard earlier in our talk this morning that 90% of drug targets fail. But one of the things that genetics actually provides is, for instance, if you use GWAS data, Eric Minical here at Broad showed that GWAS-supported drug targets and genetic-supported drug targets actually increased the relative success of drug targets in a clinical setting by almost like a factor of 2. So what I'm going to walk you through here is our current model of how do you basically integrate genetics with functional annotations. Because while genetics supports drug target discovery, I think a lot of researchers are still using traditional methods of science, which are combing through the literature, doing our own experiments, collecting all of these types of relationships to then support what they are looking at for their drug targets. So here we introduce this model Pigeon, where we try to take these gene annotations or gene sets that share common pathways, use genetic evidence from GWAS to introduce this new concept of what we're calling indirect genetic support, where genes could be supported from other high genetic evidence genes that share a functional annotation. And then we use that to actually prioritize targets. And what we see here is if you have, now that you have these two axes of direct support and indirect support, you actually can have a continuum of drug target success. And by using both of these modalities in the statistical Bayesian framework here, you can get actually really good relative success related to just even using genetics alone. So if you want to learn more, come to poster 22, and let's run some Pigeon on some of your prior knowledge and your gene sets. Thanks.